We see lots of people coming in. We'll just give everybody a few minutes to join the webinar before we get started. In the meantime, if you like, you can write in the chat where you're from and if you're part of an OWSD national chapter or what your relationship to OWSD is. Lots of people coming in. We'll just wait 1 or 2 minutes before we start. Hello, Dr. Fatima Tahir, Dr. Sadaf, welcome. Dr. Atya, welcome. Hi, Tista, nice to see you here. And the OWSD Nepal National Chapter, welcome, welcome. Okay, see lots of people still joining. Just wait 1 more minute. Dr. Saima, hello, nice to see you. Rania, Dr. Galia from Egypt. Nepal, hello everyone. Dr. Amina from Nigeria, oh, good. Some people from also other regions. Thanks for joining us. Anuradha from Sri Lanka, welcome. We have about 250 people registered today, so a very good turnout. Hello, Dr. Mapula from South Africa. More people from Africa. I guess it's also morning for you. Okay, I think we've had lots of people join, so we can go ahead and get started. I'm sure we'll still have lots of people coming in. So, good afternoon, everyone. I think it's afternoon for most of you. It's actually morning for us here in Italy, and as I said, we've seen quite a few people, at least from Africa. So, whatever time it is for you, thank you so much for joining us. We want to welcome you to this Asia Pacific region session of the Short Talks Big Impacts competition, which is organized by the OWSD Secretariat here, the Organization for Women in Science for the Developing World. We're very excited to have you with us today. This is the second edition of this competition. I don't know how many of you were able to join us last year, but we had three very exciting regional competitions and an absolutely fantastic global finale where the finalists just did an incredible job. And I'm sure that if you are able to attend, you'll agree that those presentations were all extremely engaging and impressive. Um, so today is the third of four regional competitions. This year, we've decided to make a separate competition for the Arab region, which last year was combined with the Asia Pacific region. And today, we're very excited to have 14 wonderful women scientists from across the region who will each be presenting their research in just 3 minutes. And the top two winners of today's competition will be moving on to the grand finale, which is next Wednesday, March 11th. And they will each present again in front of a panel of judges and a voting audience to determine our top global winners. And we're excited to say that the winners of the global competition will be invited to attend a scientific conference of their choice with support from OWSD. And we also might have some other surprises to announce during the global finale. So make sure you tune in for that. Uh, so we already have two winners from Africa and two winners from Latin America and the Caribbean. And today we'll discover who from Asia and Pacific region will go on to compete against them. Uh, so I will introduce myself. I'm Erin Johnson, and I work at the O Secretariat right now, mostly on the Early Career Fellowship Program and also on our awards program, as well as on communications. Uh, and today I'm happy to be with my co-chair, Tianga Talagala. But before I introduce her, I just want to give you a little bit of background about why we decided to organize this competition. Uh, so we know that it's critical to be able to communicate science clearly, especially when it comes to making research that is often very complex, accessible to different kinds of people that maybe don't have a scientific background or have an idea of scientists working away in the lab, but they don't really understand how that relates to their everyday lives or to policy issues. Here at OWSD, we have more than 11,000 members now, and we know that you're all working to solve uh, really important issues and challenges that affect your local communities and the world. And sometimes being able to communicate your research effectively is almost as important as the research itself. It can help you convince people about a particular issue or to convince communities that you work with to adopt a certain practice or to raise awareness in the public about an issue, or also to convince funders to give you money for your research. And in most of these cases, you might not have a whole lot of time to get your point across. And so we think it's important to develop the skills to describe your research and really make people understand why it's important in a very short time. So we want to focus here today on giving you the opportunity to really get to the heart of why it's important, what impact does it have, and why should people care? So that's what we're going to do here today. And now it's my great pleasure to introduce our co-chair, Tianga Talagala, who, if you were able to join us last year, you will know was actually one of the winners of the Asia region competition. And then she was the Judge's Choice winner of the global competition. And she's kindly agreed to come back this year to help us host the competition. Tianga is a senior lecturer in the Department of Statistics in the Faculty of Applied Sciences in Sri Lanka. She holds a PhD in mathematics and statistics from Monash University in Australia. And her research focuses on developing new statistical machine learning tools to help both practitioners and theoreticians make more open, explainable, and reproducible data-driven discoveries. She develops open source software tools to support reproducible research and teaching and has authored several R packages available on CRAN. She is the founder of the Dengue Data Hub, a centralized repository for dengue-related data. She is an associate editor of the R Journal, published by the R Foundation. And in addition, she is also a co-founder and co-organizer of R Ladies Colombo, the Sri Lanka local chapter of the R Ladies Global Organization, which promotes gender diversity in the R programming language. So now I'd like to hand it over to Tianga, who is going to share a little bit about her own experience last year with the competition, and she's also going to explain to us how the competition is going to run.
Thank you, Erin. It is truly an honor to co-chair this session with you. Being last year's Judge's Choice winner, this competition holds a very special place in my heart. Preparing a 3-minute talk is not easy. Every word counts, every sentence carries a weight. So first, congratulations to all our presenters for taking this courageous step to be here today and for being selected to present. Communication is very important to me, both in my teaching and in sharing my research with broader audiences. Last year, participating in this competition was truly a transformative experience. It gave me the incredible opportunity to attend the 7th OWSD General Assembly in Bogota, Colombia, where I met inspiring women scientists from different fields, policymakers, political leaders, and vice chancellors of different universities. I got opportunities to make new collaborations, gain new ideas, and many more. In addition to these experiences, the competition opened many international opportunities for me. Recently, I was invited to give a TED Talk. TED is a well-known, global, powerful platform for sharing research ideas in an engaging and storytelling format. It is an experience I would never have imagined before this competition. Today, I'm happy to be back with these 14 women who will be presenting their research in just 3 minutes. I know firsthand that this is a challenge, but it makes us think and focus what really matters. What difference does your research make in the world? So, here's how the session will run. We will give a brief introduction to each presenter, and then they will each have exactly 3 minutes to share their research with us. There will be a timer that will appear on Erin's video, which we will keep spotlighted so that the presenters can keep track of their time, but try not to worry too much about that, presenters. After all the presentations, you, our audience, will have the chance to vote for your favorite presentation based on how well you think they engaged the audience and communicated the potential impact of their research. We will be using a poll that you will see here in the webinar, so you should see that here when the time comes. Actually, you will see the poll at the end of all the presentations. I'm also excited to tell you that the top 3 winners of the global finale competition, which will be the judges' choice winner as well as the 2 top audience choice winners, will be rewarded with the opportunity to attend a scientific conference of their choice funded by OWSD. So we are here mostly to have fun and practice our science communication skills, but there are also some real stakes for today's competition. You will only vote one presentation. That means one presenter at the end. So we strongly suggest you to take notes of the presentations that you like most because there will be no voting in real time. But we will bring everyone back up onto the screen briefly before we begin the voting so you can remember everyone's names and faces. I hope everything is clear, and let's go ahead and get started with the presentations. Over to you, Erin.
Thank you so much, Tianga. It was really nice to hear about your experience, and and to learn that you are invited to give a TED Talk. That's really, really exciting, and you have to give us an update when you have that, if we can watch it, or where and when we can watch that. So let's go ahead and get started with the presentations. So again, we'll put the timer up on the screen, and we'll start it once you start speaking. So don't worry about, trying to press unmute as fast as you can, or if you have a slow connection or something. So our first presenter is Jyoti Devkota from Nepal. Nepal is a country that's very important for OWSD. We have a big national chapter there, and Nepal also hosted our first Game Changers workshop a couple of years ago there, where lots of members of the Nepal national chapter joined us to learn about how they can become policy influencers within their own institutes and communities. So it was great to get to know many members from Nepal then. Jyoti is going to tell us today about statistical analysis of the impact of micro hydropower on the status of women in rural households with examples from Nepal. And this is a very important topic because we often hear about and talk about women in science, but we hear less often, I think, about the other side of things when science and technology solutions are implemented and how this differentially impacts women and men. And that's certainly just as critical to consider. So please, Jyoti, tell us about your research.
Good afternoon, dear colleagues. The title of my presentation is Statistical Analysis of Impact of Micro Hydropower on Status of Women in Rural Households with example from Nepal. Women spend several hours each day collecting firewood and cooking on traditional stoves in many developing countries, including Nepal. Worldwide, meeting growing energy demand for development, however, is becoming increasingly difficult as fossil fuels and forest resources continue to dwindle. Thus, this challenge highlights the urgency of turning to clean, alternative, and renewable sources of energy. Microhydro project MHP is a source of renewable energy where the current of the natural rivers emerging out of mountain ranges is utilized for electricity generation. Nepal, being a Himalayan country, has thousands of rivers and rivulets emerging out of Himalayan ranges. Thus, microhydro is the main source of electricity in remote areas of Nepal. Producing energy from falling water in hilly regions of Nepal is an age-old technique. In Nepal, since centuries, using this technology, water mills with grinding stones were built. This technology is still very popular. In addition to this, in Nepal, the grid electricity is also renewable as it is hydropower based. Direct benefits of renewable energy use, especially MHP, are obvious and can be measured easily. The novelty here is that this paper demonstrates the measurement of latent variables related to the benefit of MHP to women of rural households of Nepal. Polytomous statistical models are developed here in order to explain the sensitive topic of role of wife in decision making with respect to MHP. Her importance in the decision-making process of the family is quantified using odds ratio. This is achieved by developing polytomous statistical models. A simple random sample of 110 households was selected from a population of 171 households that are consumers of 17 kilowatt Karamdana MHP located in Kavre district of Nepal. This MHP was established in 2010 in Kavre district and currently serves 171 households. It was found that women engaging only in agriculture in such households were not empowered as they had no role in decision making. But if she engages in livestock rearing, her odds of keeping profit from MHP use increases by 66269310.59 times in comparison to her husband. Similarly, if she lives in a concrete house in comparison to a mud house, her odds of keeping profit increases by 49796396.75 times in comparison to a woman who lives in mud house. Thank you very much for your kind attention.
Thank you so much, Jyoti. That was very interesting, and it seems like quite a large difference that you found. Very, very interesting results. Thank you for sharing with us.
Thank you.
Thank you. Our next presenter is going to be Vijaya Kader from India. I know we have a very large number of members from India, and I'm sure that there are many of you out there in the audience today from India. So thank you for joining us from there. Vijay is going to tell us about improving nutrition status among coastal fisher women and preschool children. So we're sort of continuing on this theme of research that benefits women, which is fantastic. So please, Vijaya, tell us what your work is doing in these communities.
Namaste to everyone. This study is supported by the World Bank actually. And here the main idea is to improve the nutritional status of the fisher women as well as preschool children, utilizing the viable technologies available in their areas. Okay. I have got 6,000 households from 28 villages across 13 districts of 4 states of Andhra Pradesh, Kerala, Karnataka, and Tamil Nadu. The results show the intake of the macronutrient is adequate, but micronutrients, especially the iron and B-complex vitamins, especially thiamine and riboflavin, are deficient. To set right this one, I have developed a sort of the amylase-rich product utilizing the germinated cereals and the legumes, and the powder was used. Feeding trials were carried out for 3 months on the preschool children as well as the women. The anemia was actually 70% in the preschool children and 60% in the fisher women. So this after feeding trials, this was really, hemoglobin content was increased. And not only that, especially the deficiencies, the clinical symptoms of the thiamine as well as riboflavin have been really corrected also. Mainly here, in this project, actually, I have fabricated two equipments. One is a low-cost ice cream freezer, where a fisherwoman can make use of the small quantity of the milk at the household level. Ice cream is liked by everyone. And another one is a fresh fish vending and display table. A fisherwoman can sit very comfortably, hygienically processing, and then preserve the fish for nearly 9 hours. And she can have a sort of the drudgery reducing of the is also main equipment. That means to improve the health status of the fisherwoman. And I have received the patent actually for these two equipment. And this was also licensed to a woman entrepreneur twice. She is handing over the check to the comptroller and documents to the register. So I was very happy, and because this was licensed twice. So, and how to improve the livelihood of, especially based on this amylase products, the villages, one NGO, they have given a flour mill as well as a sealing machine to make use of in the villages. And another one is a mushroom cultivation, was also viable technology to improve the livelihoods of the fisherwoman. So this study, and not only that, lot of literature also I have developed, mainly through this idea, technologies, that means preparation of the low-cost malt products and this, cultivation of the mushrooms. The viable technologies has really improved the nutritional status as well as the, what you can just say, the livelihood, food security, and health security. So this is the main idea of making use of the simple viable technologies. And these are all the fisherwomen actually, they are there with me. Thank you very much.
Thank you so much, Vijaya. That's obviously very impressive research, and it's great to see that there's actually, women licensing and using the technologies you've developed. It's a good evidence that it's really having a real world impact. So next, I would like to introduce Dulhari Wijaratne from Sri Lanka. Sri Lanka is another country where we have not just a lot of members, but especially a very large number of early career fellows and OWSD Elsevier Foundation awardees. And in fact, Dulhari herself is an early career fellow. So welcome, Dulhari. She's going to tell us about NK cells in dengue, friends, foes, or both. And I really like the way that you've titled your presentation, Dohari. I think it really gives it this air of mystery and makes you want to know, well, which is it? Are they friends or foes? So please tell us about this.
Thank you, Erin. NK cells in dengue, let's find out whether they are foes or friends or both. So dengue infection is a viral infection which affects around 100 to 400 million people around the world each year. And some of the individuals develop the milder form of the disease, which is known as dengue fever. But some of them develop the severe form of the disease known as dengue hemorrhagic fever. So dengue hemorrhagic fever is characterized by the severe form, the blood capillary barriers break down and the plasma, the component of blood, comes out, so the patient can even go into shock. So why it happens? Because of the patient genetics or patient immune responses or the viral factors. So I looked into the immune responses, particularly NK cell populations. So what we did, NK cells are actually the first line of defense which comes into action in viral infections, and also NK cells can kill virally infected cells in the body, and they can also produce certain cytokines like interferon gamma in patients. So what we did was we collected blood from severe form of the disease as well as milder form of the disease during the acute infection, and we looked at NK cell subsets. So CD56 bright NK cell population, which produce cytokines like interferon gamma, and the CD56 DIN NK cell subset, which have cytotoxic ability. So that means these cells can kill virally infected cells. So what we found was in severe dengue patients, right, CD56 bright cytokine producing NK cells were highly increased compared to the milder form of the disease. And they also produce a lot of interferon gamma. So if these cells are found more in dengue severe patients, these cells might be playing a harmful role, whereas the DIM population was less compared to the dengue fever, milder version of the disease. So if this is less in the severe form of the patients, these cells might be having a protective role. Why it is important? Because there are two licensed vaccines in the market, but they have their own limitations. Since NK cells are both foes and friends according to the subset, it's important to look into these subsets while developing vaccines. And it's also important to monitor these cells, how it changes according to the vaccines, vaccines. It's very important to look into these different subsets. So thank you very much.
Thank you very much, Dilhari. That was a very good reminder of how biology is never exactly as straightforward as we would like it to be. Um, so now I would like to bring on the stage Tamina Foyez from Bangladesh. Tamina is also an early career fellow, and I had the pleasure of meeting her last year in our orientation workshop here in Trieste. So it's nice to see you again, Tamina. Uh, I was also lucky enough to learn then about her research, which is about bioactive hydrogel, a multifunctional therapeutic platform rapid wound repair. Uh, but now you're all going to be very lucky to hear about it as well. So please, Tamina, for everyone that has not heard about your research, tell us about this.
Thank you, Erin. Most of us think of a word just a cut, something small, something that heals with time. But millions of people with diabetes wounds can become a silent epidemic. In the United States, about 2 million people develop diabetic foot ulcer every year. In Bangladesh, diabetes is rising quickly, and diabetes foot complications are emerging as a growing public health challenge. So why don't this wound heal? Imagine our bodies is a repairs construction crew. When there is chronic wound, the wound actually gets stuck. Our repairing mechanism gets stuck, and there is a lot of things going on. The inflammation growing like say fires, and the bacteria increase the load, and they reduce the blood supply, which reduce the oxygens and nutrients. For decades, we are using the gauze, but the gauze is a cruel healer. Dries the wound and stick painfully and often carries the infections. So now, how can we challenge these problems? We need something different. So in our lab, we developed a jelly-like things. It's a bandage, which is a biopolymer hydrogels. And these hydrogels contain the natural ingredients and 90% waters. And these gels supposed to reduce the bacterial growth and also enhance the tissue regenerations and maintains a moist environment that promotes the faster healings. Next, we ask, what if we add our own healing signals, then what happens? And here comes the platelet-rich plasma, PRP. We are thinking to incorporate the PRP with our hydrogels, and this PRP, which is full of growth factors, and this tiny messengers tell cell what to do. So when we add our own blood PRP with these hydrogels, we believe that will enhance the healing properties and instruction the cells what to do exactly. So these natural materials combined with our own blood working together to turn a silent epidemic into a story of hope and recovery. Thank you all for listening to me. And I also would like to thank my team who worked with me in this research. Thank you.
Thank you so much, Tamina. It's very interesting to hear about how you're using these materials and products together with the body's own defenses. That's really interesting. So now I would like to see if we have Dilhara Wijatungi. I know she was having a little trouble with her connection. but I think she has been promoted now to a panelist. There, yes, I see she's here. So let's go ahead and bring her to the stage. Uh, so Dilhara is also from Sri Lanka, and she's going to talk to us today about green or greenwashed, the hidden fate of biodegradable products. And this is something that I know I, as a consumer, wonder about all the time. You know, you feel better using a biodegradable fork or whatever instead of a plastic one, but it does seem maybe a little too good to be true. So please, Dilhara, enlighten us about what really happens to these products.
Thank you. Ivan. Plastics, the material once thought to be the mankind's greatest innovation, is now slowly becoming the world's long-lasting disaster. As we all know, plastics are intentionally engineered to last longer, incorporating numerous supportive characteristics like durability, low production cost, lightweight, and exceptional versatility. Plastics alone is not the big issue, but when they are mismanaged. Mismanaged plastics cause waste plastic accumulation, causing devastating impacts on the environment, wildlife, and also our own health. And apart from that, when they become little, they become even more devastating when they are broken down into little tiny pieces, such as the microplastics. Due to specific characteristics, these microplastics have the ability to seep through the surface runoff to the ocean, accumulate along food chains, and spread across continents through atmospheric transportation. Biodegradable products came into light as a promising solution for this long-lasting waste plastic accumulation because biodegradable products they have to convert into carbon dioxide, methane, water, new biomass, and energy mainly because of the enzymatic action of microbes. And these biodegradable products are available in markets under such as disposable, biodegradable, and oxo-degradable, which according to the UNEP requires different conditions when in the process of biodegradation. If we can pay attention to this photograph taken after a beach cleanup, you can see more than 90% of the debris here are plastics thrown after domestic usage. And along with this biodegradable trend, there's a huge possibility for these materials to be replaced by biodegradable products very soon. Therefore, biodegradation of such materials going to be very important when it comes to marine pollution. And in my study, in my PhD study, I investigated if these biodegradable products are working really in the local context. My test samples included a salad plate, a lunch sheet, and a garbage bag with biodegradable labels on them, and we tested their biodegradability under natural and laboratory setups. These setups represented the common dumping places in Sri Lanka, such as the freshwater riverine ecosystems, the sea, the beach, the lagoon ecosystem, the direct sunlight exposure, and microbial exposure for a period of 6 months to 1 year. But the results were shocking, since none of the test samples chemically degraded by exposing to the naturally given conditions, but some they only physically fragmented, generating microplastics. Therefore, my work exposes the misleading biodegradable labels promising biodegradation, and also it highlights that just because you throw out these just because you're throwing out this green alternative, it's not going to be biodegrade just because you throw out from your window. Therefore, we urge the manufacturers that you should include these specific conditions that are needed for your product to biodegrade in the natural conditions. And also, we urge the authorities to incorporate these experimental results in policy upgrading in Sri Lanka. And therefore, every kind of plastic should be managed carefully for a better planet. So therefore, we have to refuse, reduce, reuse, and recycle. Thank you.
Thank you very much, Dilhara. I like that you ended with like a very nice, strong call to action for both the consumers and the policymakers. It makes it very clear, what we should be doing. So now I would like to introduce Chandra Kantavelu from Malaysia. Malaysia is another very active national chapter for OWSD. We have over 120 members there, and I know they're doing lots and lots of activities there. So welcome from Malaysia. Chandra is going to tell us about empowering young women through theory-driven digital menstrual health education, evidence from a cluster randomized trial. And we know that lots of young people now access basically all of their health information online, and probably especially for something that's a sensitive topic for girls. Maybe they don't want to discuss it with parents or doctors. So please, Chandra, tell us how your work is addressing this.
Thank you, Madam Chairperson. And good day, everyone. So every month, half of our humanity bleeds, and yet we still whisper about it. We hide sanitary pads in brown paper bags, we use code words, we teach girls to be discreet, to be quiet, to just endure it. But we all know menstruation is a biological process. But the silence around it, that is constructed by socio-cultural norms. I am Chandra, a public health medicine specialist from Malaysia, and I asked a question that sounds simple, but it's not simple at all. What happens when we replace the silence with science? Not just awareness campaign, not just inspirational posters, but a structured, theory-driven, measurable health education intervention. I conducted a cluster randomized controlled trial among university students who are living in hostels. These students are young, educated, and supposedly empowered. But many still felt embarrassed about menstruation. Many practiced suboptimal menstrual hygiene, and many lacked the confidence to challenge needs because knowledge alone does not change behavior. So I designed a digital intervention grounded in the information, motivation, behavioral skills model. I named it Hide Education. Hide means menstruation in our local language, coined with education. So it became Hide Education. So it was delivered for 10 days, 10 short videos. No workshops, no product distribution, no expensive infrastructure was needed. Just structured evidence-based education to find out how it improves the menstrual hygiene practices among the students. I measured the changes post-intervention, and the results showed significant improvement in menstrual hygiene practices. Knowledge also increases, motivation also strengthened, self-efficacy grew, which tells us something powerful. When education is structured, when it is theory-driven, when it respects women as decision makers, behavior changes. Menstrual hygiene is not a small issue. It affects education. It affects mental well-being, reproductive health, work productivity, and also dignity. Across the Asia-Pacific, we speak about empowering women in science, but we cannot build confident scientists if we are still raising girls who feel ashamed of menstruation. Because when a young woman understands her body, she does not just change her menstrual hygiene practices, but she realizes her power. She shapes her future. She transforms the generations. And when women molds future generation, society transforms. That is the impact that we want. That is the real empowerment. Thank you.
Thank you so much, Chandra. I really like this evidence-driven way of approaching, education about these topics. I think it's really nice to see that that has a quantifiable impact. So now I would like to introduce our next presenter, who's also from Malaysia, Nisha Meru, who's going to talk to us about visualizing parasitic teaching models through educational artwork for Aboriginal communities. I know in our last regional session, we had many interesting presentations about emerging teething methods using digital technologies, but we haven't heard anything about using artwork as a way of teaching. And I know this will be also interesting to Tianga because her winning presentation last year was about blending art and data, which sounds kind of similar. So please, Nisha, tell us about your research.
Okay. Hi. Good evening, everyone. So we often think that the longer we speak, the more the audience will learn. But the impact is not measured by length, but is by clarity. Being in academic line for around 10 years now, teaching a STEM education subject, especially complex subjects like parasitology, microbiology, students are continuously exposed to large amount of information, life cycle of the parasites, how do they get infection, prevention, and so on. So in a classroom teaching, we often overload them with our assignments, tutorials, problem-based learning as per syllabus. But in this digital world, this information they can easily get using the AI and so on. But when will they have the real touch, aside being sent to a field work? So in my teaching work, I've been working with the Orang Asli in Malaysia, or the tribal people in Malaysia, who have a huge amount of parasite infection. So to make the students to understand in realistic how does the infection happen, and how to also to help them to spread the knowledge to prevent parasite, we actually created a teaching model whereby in that teaching model we include 3 different lifestyles. How do they get parasite infection from environment, water, and also from soil? So we had a lot of models mimicking the worm, displayed with multi-language, the tribal language, a local Malaysian language, also in English language. Creating this model, we also tested in a school. Those are taking science, and also the students give a good feedback, and also the student won prize for this teaching model. I can't display the model here because it's in the office. And moving forward, we believe that this teaching model, when directly we bring it to the tribal people, and when we explain how to do prevention of the parasite infection and to be way away from this infectious disease, it'll be very useful. Sometimes a short talk can create a long-lasting impact. Thank you.
Thank you so much, Nisha. That was really interesting. And obviously, we agree, which was the reason we're hosting this competition. Um, so I think that our next speaker on our schedule, I have not seen her actually, either in the panelists or in the audience, Shirisha Junuthulu from India. If you are in the audience, Shirisha, and having trouble connecting, just let us know and we can promote you as a panelist. But for now, we'll move on to the next presenter after that, who is Yuli Yatri from Indonesia, another country where we have a very large and active national chapter. We have over 240 members there, so it's nice to have some representation from Indonesia today. And Yuli is going to talk to us about, let me first add her to the screen. She's going to talk to us about small waste, big impact, cocoa pod husk powering clean water and clean energy. And this is one of my personal favorite kinds of research to learn about, which is using waste products to solve other problems, this kind of killing 2 birds with 1 stone. So please, Yuli, tell us about your research.
Yes. Good evening, everybody, and inspiring women in science. In this opportunity, I want to talk the topic about small waste, big impact, cacao pod husk, powering clean water and clean energy. I am a member of OWSD Indonesia chapter. I come from region where cacao grown abundantly. Indonesia is the producer number cacao in the world, beside Ivory Coast and Ghana. Until now, the problem is the cacao pod. Cacao content are 75% peel and 25% seed and placenta. If you see the peel, this is the white color is placenta, the yellow is peel or pod, and the brown is seed of cacao. Until now, the cacao peel is not exploited yet. The effect of not exploited cacao filters can damage environment. But the other side, fossil energy crisis, so energy sources and renewable are needed. For me, the abundance of the waste of cacao, this is not only the waste, but for me, the waste of cacao is potential material to be used and any material is used for the many field. I know the cacao content of cellulose and hemicellulose. This is the source of activated carbon. In my research, to change the cacao peel to be activated carbon through the process of chemical and activation carbon and chemical process and physical process to be high activated carbon and next to be used in many fields. The key of my research for me absorbent is activated carbon to be used in absorbent and to be used for the source of energy storage. From the until now, small waste maybe to be used in a potential. I start to begin with air farming cacao pod, not agriculture residue to be mixed, but they are carbon-rich biomass resources waiting to be generated. The key of my research, instead of three, the first of waste to resources, clean water solution, and renewable energy sources. I'm sure this is the potential material to be energy storage and clean water to be used in the world. From the hand of guide of the equity and sustainability, they can power clean water and enable renewable energy and redefine the communities and green transaction. Small waste can power grid energy, grid tank when science is it guide by purpose. Thank you.
Thank you, Yuli. I had no idea that either that Indonesia was producing so much cacao or that there's so much byproduct from it. So it's great that you found a solution for reusing some of that. Our next speaker is Lakshika Disanayak from Sri Lanka. And she's going to kind of keep us on this same thread of waste to wealth, talking about chemobiological upcycling of textile waste into jet fuel. And those are certainly two things that I think most people would never connect or imagine could relate to one another. So please, Lakshika, I'm curious to hear about this topic.
Thank you, Erin. Home is a special place for all of us. We love, protect, and even fight for our home. This beautiful earth is the home for all living things, and yet we are failing to protect it. Did you all know that all these clothes that you are wearing right now is a reason for environmental pollution? Textile industry is the second largest polluting industry in the world. More than 90 million tons of textile waste is generated globally every year. And of course, there are recycling systems to tackle textile waste, but still more than 85% ends up in landfills. This creates a huge burden on the environment. It causes environmental pollution, releases greenhouse gases. It even leads to loss of biodiversity and even contribute to climatic change. So that is why we put our faith on the concept of upcycling. So what is upcycling? When we consider textile waste, upcycling is using textile waste to produce high-value chemicals. For my PhD project, I'm trying to develop a chemobiological system to upcycle textile waste into a chemical that can be used as a precursor for jet fuel production. In other words, we're trying to make jet fuel from textile waste. This system is a two-step process. The first step is the deconstruction of textile waste. In our system, we use enzymes to break down complex textile waste into simple and microbially accessible chemicals. The second step is upcycling these textile breakdown products using microorganisms. In our lab, we have already developed a new bacterium as a model strain to use and upcycle waste products. So we engineered this new bacterium to use the textile waste breakdown products and produce the chemical that can be produced for that can be produced jet fuel. So this novel chemobiological system is economical and environmentally friendly while enabling green circular economy. Not only that, we believe that in the long run, this system will help to protect the planet Earth, our home. Thank you, everyone.
Thank you so much, Lakshika. That was really, really fascinating. Um, so now I would like to introduce Sadaf Ahmed from Pakistan. Uh, Pakistan is another country where we have a very engaged and active national chapter, and I've seen some people in the chat already from Pakistan here to see Dr. Ahmad. So thank you for being with us today. And Sadaf is going to talk to us about two brains under stress, protecting mother and fetus before birth. And this is a topic that it seems like we know so little about, and every day I feel like I see a headline about how pregnancy is changing mothers' bodies and brains, and we're still learning so much about this. So please, Sadaf, tell us what your research has found.
Thank you, Erin, and thank you, OSD, for giving me the chance. Today, I'm talking about a chunk of my research. So, since 2 decades, we are working on neurobiology of stress. But today, I want to share how mothers during pregnancy are exposed to psychological, psychosocial stress, and basically in low middle income countries and around the globe, maternal morbidity and mortality remains unacceptably high, and millions more women experience serious complications during pregnancy or pregnancy related causes. Among leading contributors are high blood pressure during pregnancy, that is preeclampsia, and metabolic conditions like increased sugar during pregnancy, that is gestational diabetes. But we never ask the questions that what is happening before the blood pressure rises, what changes before the blood glucose level spikes. Because when a woman is in social, psychological, emotional, and mental stress, it's not always remain environmental, it's become biological, and stress activates certain inflammatory pathway that become cellular, that make the mother's body in cellular imbalance, and that damages blood vessels and metabolic dysregulation occur. Major drivers of maternal morbidity and preterm birth and long-term health risk issues even after pregnancy, even for the both mother and child, according to the WHO, the main cause of brain health threats and everything. So in our research, what we did 3 layers of the research. One is, first, we measured oxidative stress markers biologically in all the psychologically stressed women, and we identified multiple levels of the connection. Secondly, we examined the gene expression as well, because stress can alter the major genes which has protective system for the brain health. For example, oxytocin gene, that is a critical bonding and love hormone and emotional regulatory hormone that can be altered for life in women and also the child as well. And the melatonin gene, which is supports sleep, with support anti-anxiety impacts, it would support neuroprotection, which support vascular health. So when these systems are disrupted, the maternal brain become more vulnerable throughout the life. And third, we look beyond delivery, how the circumference of the fetus has changed, how the fetal brain and the cognitive impact has made throughout the life. So as prenatal stress exposure is linked to the altered stress reactivity, increased risk of anxieties, metabolic dysfunction, neurodevelopmental changes later in life, means that maternal stress is not just physiological experience, it is about the two brains. Yet most of the maternal clinics, we found out, only measure blood pressure, only measure glucose. They don't have these stress measuring tools. So our team, after finding out this, I'm very proud that we developed a culturally adapted psychological stress framework, and that actually leads to the fetal programming effects, how we can control it in a very low cost, like yoga, like mindfulness, because maternal mental health is not luxury, it is a physiological need. Thank you so much.
Thank you so much, Sadaf. I really like how you drew this very clear distinction between the physiological and environmental impacts and the actual biological and psychological impacts, which are not so separated as we often think. So next, I would like to invite Hoai Nguyen from Vietnam onto the stage. And Vietnam is a country where we actually do not have a national chapter. And in fact, we only have 7 members in Vietnam, and we would love to have more. So maybe we can talk later, Hawaii, about how to start a national chapter there. But in any case, we're very glad that you've decided to join us today. And you're going to tell us about understanding antimicrobial resistance and developing antimicrobial alternatives. And this is another topic that it feels like we hear about all the time in the news, and it seems sometimes very scary as someone who doesn't have a very good idea about what kind of solutions are out there for this problem. So please tell us about some of these alternatives.
Good day, everyone. I am Dr. Hoai, and I lead the Medical Microbiology Research Group at the Research Center for Infectious Diseases, International University, Vietnam National University in Ho Chi Minh City, Vietnam. And for more than 10 years, my team and I have been working to understand and mitigate the antimicrobial resistance, or AMR in short. And AMR happens when bacteria no longer respond to antibiotics. Looks less dangerous. This is not a future problem. It's happening now. That's why you listen to that on public media all the time. It happened in the hospital, on farms, and also in our communities. And AMR is named widely by the scientific community as a silent pandemic, which brings immense danger to the community. So we study AMR development in 2 major pathogens, Staphylococcus aureus and Pseudomonas aeruginosa, which cause serious infections. One important discovery from our research is this: AMR is not caused just by one bad gene. Bacteria use a whole network of genes to survive, and many of these genes control how bacteria produce and use energy, their metabolism, rather than how aggressive they are. In simple terms, bacteria survive antibiotics by reorganizing how they function internally. Understanding this helps us think about new ways to treat infections more efficiently, not just by targeting one gene, but by disrupting the survival system of the bacteria. The research should not stop in the laboratory, it must help people. So we developed practical solution. We created a rapid, affordable test to detect Pseudomonas aeruginosa in the environment. This land-based test can be used outside advanced laboratory, help community monitor waste, water, environmental sources before infection spread. We also expand our work into agriculture. My team successfully transformed agriculture waste into special carbon nanoparticles that kill harmful pathogens. These innovations aims to reduce the needs of chemical fungicides, support safer and sustainable farming, promote cycling of agriculture byproducts, and help protect crops and food security. AMR is really a complex challenge, but at our center and our team specifically, we are turning scientific discovery into real solutions that protect communities. That is our mission, and it is what my team and I work on every single day, turning science into solutions that protect our communities. Thank you for being part of this journey with us. Thank you very much.
Thank you so much, Hoai. It's very reassuring to know that there are people like you out there working on this issue. Next, I would like to introduce Ugiloy Yusupova from the Republic of Uzbekistan. Uzbekistan is part of our Central Asia national chapter, along with Kazakhstan and Kyrgyzstan, and we're really glad to also see some representation from that region today. So Ugila is going to tell us about From Plants to Patients, phytoecdysteroids, I'm sure that I didn't pronounce that right, but you'll correct me, in modern therapeutics. And yes, I have to admit I had to Google what that even is because I had never heard it before about phytoecdysteroids, but it seems like a very interesting group of compounds. So please tell us more about that, Ugilay.
Hello, everyone. Let me ask you a simple question. Have you ever felt tired, not just physically, but deeply tired because of long-term stress? Stress is today everywhere. It affects our minds, our metabolism, most particularly our immune system. When our immune system becomes weak, our body struggles to protect us. So imagine our immune system is as a shield. It protects us any damage from outside, from viruses, infections, yes? So my research focuses on one important goal. How can we help the body stay strong under stress? Surprisingly, the answer may come from plants. Plants cannot run away from harsh environments, yes? They cannot escape another way from drought, heat, or biological threats. They survive by developing internal protection system. Some of these protective compounds are called phytoecdysteroids. Look at the picture and see them as nature's stability molecules. In humans, I always study with my colleague, with my students, how these similar compounds, this structure may help the body maintain balance during stress. Without disturbing natural hormonal system. And testing this compound randomly to the animal, my mouses, rats, rabbits, yes, we use preferred computer tools, machine learning, to predict which version, which this structure may help the body, may work the best. This allow to create better natural inspired molecules. Additionally, it reduces the need animal experiment. At the same time, unnecessary chemical experiment before even enter the laboratory where it's doing something by hand, yes? In other words, we use technology innovations to learn from nature more efficiently. And why is this important today? Because around the world, stress-related disorders, diseases are increasing, and many communities don't have, unfortunately, access expensive treatments. Moreover, adults, children, kids cannot take pills because of synthetic drugs. If we can develop safer, accessible, and plant-inspired solutions, we can help strengthen health resilience globally. And don't forget, resilience is not luxury. It's necessity for our future. That's all. Thank you very much.
Thank you very much, Ugula. It's very interesting to hear. I mean, I know that scientists have been using inspiration from plants for forever, basically, for therapeutic treatments, but it's interesting to hear how machine learning is accelerating and making that more efficient. So now we have our last presenter, last but not least, who is Zubia Naqvi from Pakistan. And she is going to tell us about genetic alterations and clinical patterns in brain tumor patients, a case control study. And I think that our knowledge about tumors and cancers has come a long way, but there's still so much to discover. And especially brain tumors have a very high mortality rate compared to some other kinds. So this is, of course, critical research. So please, Zubia, tell us what your research has found.
Good afternoon, everyone. I thank you, Erin, for introducing me. And I serve as the Events and Media Coordinator for the OWSD Pakistan National Chapter. I'm also working as a research assistant at an engineering university. So today I'm going to talk about my MPhil research, which was based on brain tumors. And Pakistan has in Pakistan, brain tumors rank as 11th most common cancers with a 5-year survival rate. So what happens is when patients are diagnosed with the most aggressive forms, they die within 6 months. And the low-grade tumors often go undiagnosed. Globally, researchers have been working on imaging tests, diagnostic procedures, therapeutic treatments, but the genetic mechanisms that underlie brain tumors are still not fully understood. So this is what we worked on. And to explain our approach, I'd like to show you something very simple. This is a simple sentence. Now, if I replace this letter I with an A, this changes the entire meaning of the sentence. Now, just like a sentence, our DNA also has nucleotides. And if there is a change in nucleotide, it changes significantly in the cellular processes. So what we worked on was a transcription factor CREB that is highly expressed in the brain, and it acts as a converging point for different signaling cascades. So it controls over 4,000 different genes that function in cellular processes. And now, if a disruption occurs in the CREB, what happens is it disrupts the entire signaling cascade. That leads to tumor development. What we did was we analyzed patients' blood samples and their clinical data as well. We used computational and molecular tools for this, and we correlated the clinical pattern with the anthropometric profiles, and we looked for mutations in the functional region of the CREB gene and in one of its downstream targets that was related to cell survival. So this contributes to foundational genomic data in Pakistani population, and it also serves as a basis for early molecular detection, risk stratification, and eventually targeted intervention therapies such as personalized medicine. So instead of treating every patient the same way, we can treat them based on their genetic profile. Because sometimes understanding a single letter in our DNA can make us rewrite someone's future. Thank you.
Thank you so much, Zubia. I really, really liked the example that you gave with the sentence about the cat. I think I've heard this concept before, but I've never seen it exactly expressed like that, and it really makes it very clear. So thank you. Uh, so now I would like to bring back on Tianga.
Okay, thank you so much to all of our presenters. Listening to these wonderful speakers, I learned so much from them. Through their talks, it became very clear to me that their work is not just a career. I truly realized how much passion they have for their work and how deeply committed they are to their research and the communities they serve. The presentations covered a wide range of topics. Each presentation showed how research can move beyond theory to make a real-world impact. The future of science is not just in discovery; it is in service. I sincerely thank OWSD for giving us this incredible platform and visibility for our work. It means so much to me, and I'm sure all the speakers share the same feeling. An opportunity like this truly motivates us to continue our research and make a difference. And now we heard all 13 presentations. It is time for the audience to cast your vote for the best presentation. But first, we are going to bring everyone back up on screen briefly so you can see their names and faces again. So let's bring everyone back on screen.
Thank you, Tianga. So first we had Jyoti Devkote from Nepal. Let's see. I'm just going to ask you all to start your videos if you don't have them on so we can just see you again quickly. let's see, that's Jyoti, who talked to us about the impact of the micro hydropower on women in rural households in Nepal. Then we heard from Vijaya Kader from India, who talked to us about improving the nutrition status among coastal fisherwomen and children. Then, where is Duhari? Then we heard from Dulhari Wijaratni from Sri Lanka, who talked to us about NK cells in dengue, whether they're helping or harming dengue or both. It seemed that the answer was both. Then we have Tamina Foyez from Bangladesh, who talked to us about developing a bioactive hydrogel for more rapid wound repair. Then we have Dilhara Wijatunge from Sri Lanka, who told us about how she's investigated the fate of biodegradable products and whether this is a real benefit or if it's only greenwashing. Then we heard from Chandra Kantavelu from Malaysia, who talked to us about empowering young women through theory-driven digital menstrual health education, Malaysia. And then we heard from Nisha Meru, also from Malaysia, who talked to us about teaching models to teach about parasites to Aboriginal communities through artwork, through educational artwork. Then we heard from Yuli Yatri from Indonesia, who talked to us about recycling cocoa pods into products to help produce clean water and clean energy. And then we heard from Lakshika Disanaye from Sri Lanka, who also talked to us about waste to wealth, about recycling textile waste into jet fuel. Then we heard from Sadaf Ahmed from Pakistan, who talked to us about the stresses on maternal and fetal brains of pregnancy and how we can protect against those. Next, we had Hoai Nguyen from Vietnam, who talked to us about antimicrobial resistance and some alternatives that her team is developing for those. Let's see, do we have her? There she is. Thank you. Then we heard from Ugiloy Yusupovo from Uzbekistan, who talked to us about developing therapeutic benefits from plants. Thank you, Ugiloy. And last, we heard from Zubia Naqvi from Pakistan, who talked to us about genetic alterations in brain tumor patients in Pakistan. I think that is everybody, unless I've missed anyone. Please say so. So we can now go ahead and open the poll. And we'll keep the poll open for about a minute and a half, 2 minutes if we see people are still voting. So please take a moment to vote for the presenter who you believe communicated their research most effectively and who did the best job at showcasing the impact of their research. And don't forget that the top 2 presenters will move forward to the grand finale. So your vote helps decide who is going to compete next week against the other regions. So we'll go ahead and open the poll and we'll see you back here in just about 2 minutes. And you should now see the poll. Okay, just a few more seconds to get your votes in. Okay, I don't see a whole lot of people still voting, so we'll go ahead and close the poll and just give us a few minutes to make sure we've got the right winners calculated. Okay, Tanga, are you ready to announce the winners? I've sent you a message, I think, so we have the right order.
Let me check the message. Is it in chat here?
Yes. Yeah.
Let me check the. I cannot see it, Dareen, unfortunately. Would you mind doing it?
Yes, sure. No problem. I'm happy to announce the winners. So, after careful voting from you all, we are pleased to announce the top four winners because we had a three-way tie for fourth place, so we won't do a fifth place. But in fourth place, we had a three-way tie between Jyoti Devkota. It's Jyoti. There she is. As well as Lakshika Disaniye. And Zubia Nakvim. So congratulations to the fourth place winners. Very well done. Then in third place, we had Dulhari Wijaratni from Sri Lanka. Congratulations, Dulhari. Very well done.
Thank you. Thank you.
In second place, we have Tamina Foyez from Bangladesh. Congratulations. That means you'll be moving on to the grand finale next week, which is very exciting. And in first place, we have Sadaf Ahmed from Pakistan. Very well done, Sadaf. Thank you very much. Oh, I think you're muted. Let's see. Yes. Yeah, now you're
Thank you.
unmuted. Yes. Excellent. Thank you very much. So thank you so much to all of our amazing presenters. I think, I hope that it was fun for you and that you all felt like you learned something and got to practice your science communication skills. And we look forward to seeing Tamina and Sadaf next week for the grand finale. Uh, so And thank you very much also to our audience. We have such an engaged and vibrant community here at OWSD. as today's turnout has shown. Thank you to Tianga for being an excellent co-chair and sharing your, inspiring words with the presenters earlier. And I think, we've really seen today how science can make a difference and how important it is to communicate that to the world. So if you're interested in joining us tomorrow, we have the last of the regional competitions for the Arab region, which is going to be starting at 2:00 p.m. Italy time, which is Central European time. And then definitely make sure that you register for the grand finale competition next week, March 11th, Wednesday. And if you're an OWSD member or you're subscribed to our mailing list, you should see that registration link in your inbox probably tomorrow. So we'll see Sadaf and Tamina next week, and hopefully many of you in the audience as well. And we wish you a wonderful afternoon, evening, and we hope to see you next week for the finale. I don't know if you'd like to add any last words, Tianga.
Yeah, congratulations to everyone. That's a wonderful session, and looking forward to see the winners at the grand finale. Thank you, everyone. Good luck.
Thank you.
Thank you.
Thank you, everyone.
Thank
you.
you.
Bye.
Thank you very
you.
much. Thank you very much.
Thank you very much.
Thank you.
Thank you.